Lecture 7 will be about the Human Genome Project, which produced the first nearly complete sequence of the human genome. But first I want to talk about two technologies that made the Human Genome Project possible. One, PCR, and two, DNA sequencing. PCR, or polymerase chain reaction, is a method for using in vitro replication in a test tube to amplify the amount of DNA material. This is how, in shows like CSI, that forensic scientists can take a small amount of blood and amplify the DNA in that blood so that there is enough to sequence to identify the source of the blood. PCR involves repeated rounds of DNA replication in a test tube, with each round doubling the amount of DNA. The DNA replication reaction is primed by DNA primers, which determine which region of the DNA is amplified. This animation provides a good description of the PCR process, and you should take 5 minutes to watch it. Welcome back. In summary, what PCR does is amplify the amount of DNA from a quantity that is too small to analyze to a quantity that can be analyzed. Not all the DNA is amplified. The region of amplified DNA is specified by the DNA primers. After the amplification, you can analyze the size of the product or the DNA sequence of the PCR product. This technique has revolutionized molecular biology because often DNA extracted from a sample is very limiting in quantity. The second key technology is DNA sequencing. Using DNA sequencing, you can determine the sequence, the string of A's, G's, C's, and T's of any region of DNA. I will briefly run through the conceptual basis of the Sanger method of DNA sequencing, named for its inventor, Fred Sanger. The first key concept is that there is a special nucleotide called a dideoxynucleotide in which the 3' hydroxyl group is replaced with a hydrogen atom. As a result, the nucleotide can be incorporated into a newly synthesized DNA strand, but the strand cannot be extended because the next nucleotide is supposed to form a phosphodiester bond with the missing 3' hydroxyl group. As a result, the dideoxynucleotide terminates the DNA synthesis. If you spike your DNA synthesis reaction with a small amount of dideoxynucleotides, then at some low frequency, they'll be incorporated into the newly synthesized DNA strand and terminate the synthesis of that particular strand. The key point is that the DNA sequence of the template strand determines which of the 4 dideoxynucleotides will terminate at a specific position, resulting in a fragment of a specific length. These DNA fragments of different sizes, terminated by one of the 4 dideoxynucleotides, can be separated by size on a gel, resulting in a ladder. If each of the dideoxynucleotides are labeled with a fluorescent probe of a different color, then we can determine which dideoxynucleotide terminator corresponds to each of the DNA fragment lengths. And from this we can read the DNA sequence. For example, if the A dideoxynucleotide is green, and G is black, C is blue, and T is red, then we can read the sequence from 5' to 3'. In this example, it is T, G, C, G, T, C, C, A, C, and so on. In summary, I can take a sample of my saliva, which contains a small number of cheek cells, extract the small amount of DNA, and then amplify the DNA by PCR so that I have enough to sequence. And then I can use DNA sequencing to obtain the nucleotide sequence of any region of that DNA. In this way, it is theoretically possible to sequence my whole genome. And that was the goal of the Human Genome Project, to sequence the human genome for the first time. It was started in 1990 and the rough draft sequence was completed in 2001. One key innovation that made this project possible was the development of faster and faster DNA sequencing machines that used the Sanger method. These machines carried out the DNA synthesis reaction with the fluorescent dideoxynucleotides and then ran the products on a gel where the latter fluorescent fragments could be read and processed by computers. Previously, all this work was done by hand. One limitation, however, was that only approximately 1,000 base pairs, or 1 kilobase, could be read from a single reaction. So it was as if you're trying to read a very long book containing billions of characters, 1,000 characters at a time. The way around this problem was called shotgun sequencing, in which random 1,000 base pair regions were sequenced and then assembled into longer sequence segments based on overlapping sequence reads. The drawback was that this required that each region be sequenced multiple times. In 2001, after a very laborious process, about 90% of the sequence, termed the rough draft of the human genome, was obtained and published. The remaining 10% was sequenced for the next few years, but was difficult to obtain because it was in regions of repetitive DNA which are hard to sequence. The total cost of the project was over $3 billion. A vast amount was learned from the first human genome sequence. The haploid human genome contains approximately 3 billion nucleotides. There are approximately 20,000 genes split among the 23 haploid chromosomes. About 1.5% of the genome are protein-encoding exons. About 20% are introns between the exons. Another 75% of the genome are the intergenic regions between genes. Some of these intergenic regions contain transcriptional regulatory sequences for the regulation of gene expression, but most is of unknown function. Indeed, the 95% of the DNA representing intergenic regions, introns, repetitive DNA, and so forth are sometimes termed junk DNA because their function is not known. The long-term goal is to be able to sequence a human genome for $1,000. At that price, it becomes feasible to sequence everyone's genome. At the very least, we can sequence the genomes of all 5 million newborns each year for the total price of about $5 billion a year. As you can see, the price of sequencing a human genome is coming down rapidly. It cost $3 billion to sequence the first genome. But once we had the first reference sequence, it became much easier to sequence the next genome using the reference sequence as a guide. So in 2001, the cost of sequencing the next human genome was not $3 billion, but approximately $100 million. Since then, the cost has been plummeting. Notice that the y-axis is actually on a log scale. Today, the cost of sequencing your genome, or my genome, is about $5,000. In 5 years, it should drop to $1,000. Once the cost drops down to $100, then everyone will have their genome sequenced. The key technological advances making this cost reduction possible have been more efficient robotics for preparing the DNA sequencing reactions and more highly parallelized sequencing. So the day is rapidly coming when everyone will have their genome sequenced. Everyone will have their own book of me. For assignment 7, I want you to further develop this idea, this metaphor of the book of me. This is important, and so I want you to spend approximately 1 hour on this assignment. First, create a Google document named Assignment 7 followed by your name. Share this document with me. The first question is, how many characters are in your book of me? The second question is, how many chapters are in your book of me, and what does a chapter represent? The third question is, how many paragraphs are in your book of me, and what does a paragraph represent? For the second half of the assignment, I want you to think about the definitions we went over in video lecture 1 and what they correspond to in the book of me. What represents chromosomes in the book of me? What represents genes in the book of me? What represents alleles in the book of me? What represents mutations in the book of me? What represents a genotype in the book of me? Please explain your answers. It's important to note that there is not necessarily one correct answer. I am much more interested in your reasoning to justify your answers. When you're done, copy and paste your answers. When you're done, first make sure you've shared the document with me, and then copy and paste your answers under question 15 of your term paper. In summary, PCR makes it possible to amplify a small amount of DNA to a much larger quantity that can be sequenced. DNA sequencing produces the DNA sequence of a region of DNA and is largely performed these days by automated machines. The Human Genome Project produced the first human genome sequence. Since then, the cost of sequencing human genomes have dropped precipitously from approximately $100 million to about $5,000 today. Once it reaches $1,000 or below, it will become feasible to sequence everyone's genome and produce a book of me for everybody.